In this experiment, we investigate the gas produced during photosynthesis. As you recall, oxygen gas is a byproduct of photosynthesis, and this production of oxygen is easily collected from aquatic plants such as the pondweed, which is commonly referred to as the elodea. Other plants that may be used include the spirogyra and the water lily. Now, given that water usually has a low concentration of carbon dioxide, the water is enriched with CO2 through the addition of sodium hydrogen carbonate, which dissolves to release carbon dioxide. So the addition of sodium hydrogen carbonate boosts the amount of carbon dioxide in the water so as to promote the process of photosynthesis. Now we also make sure that there is adequate light, because light, just like carbon dioxide, are factors that are required in order for photosynthesis to take place. So the setup is put up like this diagram here, and then, as I mentioned, you make sure that it is exposed to sunlight to allow photosynthesis to take place. Now we leave the setup in the sunlight until sufficient gas will have collected in the boiling tube. Then we test the gas using a glowing splint. We test the gas using a glowing splint, and if the gas is oxygen, then the glowing splint will relight or will glow brighter. So from there, we will conclude that the gas produced during this experiment is oxygen. Now, this experiment can be used to investigate various factors on the rate of photosynthesis. In other words, we can use this setup to investigate various aspects of photosynthesis, and this includes the effect of light intensity on the rate of photosynthesis. By using a light source, so we modify the experiment slightly. So we provide a light source. This light source, instead of sunlight, we get light energy from this bulb. Now, since we do not want temperature to be a factor in the experiment, we do not want temperature to vary, there's a special heat screen that we use to prevent the heating up of the setup by the bulb. Otherwise, the setup remains just like in the first one. Here we have water that is enriched with sodium hydrogen carbonate. We use an aquatic plant like elodea. Now we can vary light intensity by varying the distance between the lamp and the plant. The longer the distance, the lower the light intensity. And the shorter the distance, the higher the light intensity. So rate of photosynthesis can then be recorded and compared, because you can determine the rate by counting the number of oxygen gas bubbles being produced per unit time. For example, you can count the number of bubbles per minute at each distance. So let's say you can start, if the distance is 15 centimeters, we can count and record the number of oxygen bubbles produced per minute. Then we move the distance further to say 20 centimeters. Then we count and record the number of bubbles of oxygen produced per minute. So you keep varying the distance so that from the data we'll be able to relate the distance, which determines the light intensity, and the rate of photosynthesis measured by the number of oxygen bulbs produced per minute. So the same experiment can be used through modification to investigate the effect of carbon 4 oxide concentration on the rate of photosynthesis. For example, by using known quantities of sodium hydrogen carbonate, and therefore we know an estimation of the concentration of the CO2 in the water, we can then determine the rate at which oxygen is being produced by keeping the light intensity constant and also the temperature constant. So we can vary the concentration of the CO2 and then measure the rate by counting the number of oxygen bubbles being produced. We can also investigate the effect of temperature by placing this setup in different temperatures and counting the number of oxygen gas bubbles being produced at a given temperature. Then from here we can relate the effect of varying temperature on the rate of photosynthesis, just like we can relate the effect of different concentration of CO2 on the rate of photosynthesis. So this experiment is very important because we use it to measure or investigate the various factors and how they affect the rate of photosynthesis. The main factors that we use or that we investigate include: one is the light intensity, temperature, and the concentration of carbon 4 oxide.